Hello, everybody. I'm Andrew Shang. This is my presentation for USRG 2021. My project is Protein Sequence Embedding for Binary Prediction of Protein-Protein Interactions, which falls under the NSF grant number 1943008. I've been working under Dr. Yang Shin at the Electrical Engineering Department here at Texas A&M University. So for this presentation, I will break it down into several main sections. I will first provide an introduction on proteins, the objectives of this project, current methodologies, as well as our proposed solution. And finally, I'll be discussing my results and any future directions to take with this project. Now, proteins. Proteins are large biomolecules and macromolecules which perform a vast array of functions within organisms. So if we take a look at this image on the left, we see a structure of myoglobin, which provides oxygen to working muscles in your body. Aside from myoglobin, there are numerous other proteins that function with different capabilities, including catalyzing metabolic reactions, DNA replication, responding to stimuli, providing structure to cells and organisms, and transporting molecules from one location to the next. Now, what is the very simple definition of a protein? A protein is just a large molecule made up of one or more long chains of amino acid residues bonded together by peptide bonds. It can really easily be thought of as a long chain of Legos connected together. If we just take a look at this image right here, we see multiple different colored beads, and each bead represents one amino acid. And connecting each amino acid is a black bar. That black bar represents the peptide bonds. And proteins can only be formed by a set of 20 amino acids. That can be similar to maybe a dictionary. And all those are represented right here in this table below. Now, at its primary structure, it looks extremely similar to a string of letters or maybe just a sentence in the English language. And as a protein increases in complexity from primary protein structure all the way to quaternary protein structure, it starts to look a lot more sophisticated as it folds together. Now, so how does a protein compare to technology? So a laptop or phone, it also has different levels of complexity. There's circuits inside, a motherboard, a CPU, and some RAM. And at each level, it increases in complexity for its various functions. And at each small portion, it does its own task. With code, it works the same way. Small lines of code slowly grow to form into larger, complex programs. So currently, there are a lot of experimental methods for discovering protein-protein interactions. Most common is a principle-driven solution that's named protein docking. Now, protein docking is the prediction of the structure of a protein-protein complex from the structures of the individual proteins. Now, the issue with protein docking and any other similar methods just results from its time-consuming and labor-intensive nature. Additionally, there are a lot of knowledge gaps in the three-dimensional space of protein-protein interactions, which leads to inaccuracies in the experimentally derived results using protein docking or any other similar method. Now, for our proposed solution, we actually take a data-driven deep learning method to predict protein-protein interactions. Numerous databases such as UniProt and PDB have accumulated protein data gathered experimentally. This allows for direct computational methods of predicting PPIs. This has a very clear benefit in being inexpensive, time efficient, and applicable to any proteins without any physical limitations. Now, there are a few embedding architectures that we used for protein-protein prediction. Now, this table gives a breakdown of varying combinations of embeddings along with its corresponding downstream and predictive modules. So the first we see here is HRNN, second is CNN, and the third is transformers. So for the downstream model of all three embeddings, we use ReLU, sigmoid, linear ReLU, and a BCE loss. And for its predictive modules, we use a fully connected layer and a softmax. Now, we're first going to discuss the embedding using an HRNN or a hierarchical recurrent neural network. Protein sequences can be represented as 1D modality text sequences with a label describing whether the inputs are interacting, so a 1, or a 0 for non-interactions. Now, this is all fed into our model that has an HRNN embedding, which is designed to model hierarchical structures in sequential data, which perfectly fits a protein. Now, the output would be a two-dimensional probability vector, one for interacting and the other for non-interacting. And both of these will sum up to 1. So that's our first embedding architecture. Our second embedding architecture will be called a CNN. This is also known as a convolutional neural network. where we use a convolutional embedding, which is mostly used for classification and computer vision tasks. Now, like with the HRNN, the input is still the same of any two protein sequences with its label of 1 or 0, and then the output is still the probability vector, which the two values should sum up to 1. Now, for our third embedding architecture, we use a transformer, which this transformer uses attention mechanisms to capture global patterns effectively without the use of any RNNs, such as the HRNN that we previously shown. Again, likewise with the other two, the input is the same and the output is the same. Now, what is the performance evaluation for our PPI interactions? For the HRNN embedding, these were all run after the hyperparameter values were fine-tuned to be as optimal as possible. And the dataset was curated with a custom dataset that we provided. Here, we both use AUPRC, which is area under the precision recall curve, and AUROC, which is area under the ROC curve, as the two performance metrics. Here, to describe how to understand this graph, we just want to know that the curve is as steep as possible, which both these two graphs show. That means there actually is a very high accuracy. Now, the formulas above are just some of the formulas that we use to calculate this. So this is for HRNN. Now, what about for CNN? So these two graphs right here are for the CNN embedding, where the graph on the left is the AUPRC for the training set and the test set. From here, there's actually a slight decrease that we can notice in right here and right here, the two steepness of the two graphs, where from the HRNN, it might be steeper, but in the CNN, it's actually less steep. Well, it's still a relatively high accuracy. It's still noticeable the difference in accuracy that a CNN provides. Now, the last embedding method is the transformers. So for the transformers, the two training and test set graphs show an extremely high accuracy. We notice this because we see right here, the two graphs have a very steep incline. Now, this method seems to have the highest accuracy between the HRNN, CNN, and transformers, which this falls under our prediction, as transformers are a current best tool borrowed from the field of NLP, or natural language processing, to find any global patterns. Now, to summarize, a high accuracy was achieved to predict protein-protein interactions. It could be seen from the various representations of HRNN, CNN, and transformers that transformers yielded the highest accuracy based on the steepness of the curve. Now, for any future considerations with this project, multimodal input data of residue-residue contact maps and multitask learning techniques to predict continuous geometries and angles can be implemented. And this concludes my presentation. Thank you very much.